Hello. Today, we're going to talk about classification algorithms. This is a kind of supervised learning in which you have categorical output. For example, in the introduction, we introduced the problem of trying to diagnose cancer from images of cells. Here, the output is a categorical value. Either the cells are cancerous or normal. First, we'll go over different applications of classifiers in biology. For example, there's many biomedical applications for classifiers. For example, each observation might be a different patient. Our aims might be to develop diagnostic tools to predict the best personalized medicine for the patient. And then, once we build such models, we could then look at those models and try to identify biomarkers to help with diagnosis or biomarkers that help decide which personalized medicine to give each patient. Our input variables could be different physiological features of the patient, such as its age, height, weight, blood pressure, etc. It could be molecular factors, such as genotype, its transcriptome, proteome, metabolome, etc. Or it could be sensor data, image data, audio data, etc. The output of the model could be predicting a category, such as disease or healthy. It could also be a different type of categorical phenotype, like eye color. In the case of personalized medicine, we could try to identify which drug would work best on this patient. There's other biomedical applications where the observations are potential drugs. In this case, the aim is to identify which of the potential drugs are most promising. The input in this case would be different aspects of the chemistry of the drug or different outputs of experiments of the drug in different tissue cultures, such as how the drug impacts the phenome, the transcriptome, the proteome, etc. The output would be a label of whether the drug was successful or not successful. There are also ecological applications of classifiers. For example, we might want to develop a tool that will help us automatically track animals. So in this case, the input data could be satellite images, motion capture images, or even images captured by citizen scientists. Audio could also be an input, such as nature recordings. And the output would be the species that is in the image or in the audio recording. In addition, we might want to use classifiers to predict future ecological phenomena. For example, we might want to predict things such as which species of grass would dominate the next year. Will eggs be laid on this beach? Yes or no? Will there be algal blooms here in 6 months? The input data could be various genetic data, such as metagenomics data, or it could be water and soil chemistry or other sensor data. There's also lots of applications of classifiers in fundamental biology. For example, we might ask, will a transcription factor bind to a specific DNA fragment? In this case, the input would be a regulatory DNA sequence, and the output would be whether the transcription factor binds or does not bind. We can also ask, will an RNA sequence form a certain kind of secondary structure? In this case, the input is an RNA sequence, and the output is the RNA secondary structure category. We can ask, which proteins are extracellular markers? The input is in the protein sequence, and the output is extracellular marker or not extracellular marker. We can ask, which organ will a specific gene be expressed in? The input could be the regulatory DNA sequence, and the output would be the organ that the gene is expressed in. We could ask, what are good biomarkers for cell type? In this case, we try to build a model that predicts cell type for maybe something like a single cell transcriptome. And then we'd look at the model itself and try to use that to identify good biomarkers. We could also use classifiers as a tool for doing things like automatic image labeling. So for example, we might want to input an image and as an output have what's in the image. And this could be helpful for things like automatic labeling of microscopy images. Today, we're going to talk about general principles of classifiers and ways of building specific classifiers, such as support vector machines, random forest, and logistic regression. Finally, we'll briefly talk about variable importance. So what counts as a good model? One way is percent accuracy, which is the number of correct over the total number times 100%. However, we need to always compare our percentage accuracy to a proper baseline. For example, let's say that in our real data set, 80% of the time the cells are normal cells and 20% of the time the cells are cancer cells. I could make a really silly classifier that always predicts normal. This classifier would be 80% accurate because the data isn't evenly balanced between cancer cells and normal cells. Accuracy is still commonly used. However, I just use this as an example to illustrate how important it is to always compare accuracy to a proper baseline. There are other important metrics for evaluating a model. For example, you might want a high true positive rate and a high true negative rate, while also having a low false positive rate and a false negative rate. Deciding the best model is often a balancing act. For example, a general practitioner would probably want a diagnostic test with a low false positive rate because they won't want to send a lot of people to specialists unnecessarily. Specialists might want to test with a high true positive rate because it's more likely that the person who's already gone to the specialist has the disease. Often, when you build a model, there'll be a single parameter that will determine the balance between these metrics. One way of depicting this balance is with receiver-operator curves. In receiver-operator curves, the x-axis is the false positive rate, and the y-axis is the true positive rate. You can change the parameters of the model and each time calculate the false positive rate and the true positive rate on your testing set. And from this, you can draw an ROC curve. If the model was completely random, then you'd expect a line like this. A pretty good model would look like this, curving upwards, because this would indicate that the true positive rate would grow much faster than the false positive rates. A really good model would look like this, indicating that you get a really high true positive rate while having a very small false positive rate. This can be summarized by the metric, the area under the curve. In the random example, the area under the curve is 0.5. This is because the total area of the square is 1, and the area under the curve is half the area of the square. In the perfect model, you'd get the whole area of the square, which is 1. Next, we're going to describe specific ways of building a classifier, such as support vector machines. First, I want to remind you how the data is structured. We imagine that each row is an observation and each column is a variable, and we have one column of categorical output variables. We can visualize the data with a series of scatter plots. For example, we could plot variable 1 versus variable 2 and have each observation be an X. We could color code those observations based on the category. When I explain support vector machines, I'll start off by explaining it as if there were only two input variables. In support vector machines, we draw a line, and everything above the line is labeled as being part of one category, and everything below the line is labeled as being part of the second category. But what counts is the best dividing line. We want our model to be as accurate as possible. That means that this line would be bad because it would unnecessarily include this red X in the same category as these blue X's. So it would be less accurate. In addition, we want the 2 points that are closest to the line, which in this case is this point and this point, to be equally spaced from the line. So, for example, this wouldn't be as good of a line to separate the two categories as this line. This is because if we have new data and we observe that this new data is over here close to these blue points, we'd want that data point to be labeled as a blue data point, not as a red data point. Also, we want the two data points that are closest to the line, in this one and this one, to be as far away from the line as possible. For example, this wouldn't be as good of a dividing line as this one. However, this is a little bit of a balancing act. So, for example, this line would be as accurate as possible because you get 100% separation between the red X's and the blue X's. However, let's say that we have a new data point that's over here where there's the circle. You might think that that circle is probably more likely to be red than blue. So this might be overfitting to the data. Drawing the line like this means that the separation isn't so dependent on this outlier. And it could be that this model is more generalizable and less overfit. Sometimes you need to transform the data before doing support vector machine. For example, you can clearly see that the red points and the blue points are found in different areas of the graph. However, you wouldn't really be able to separate out these two classes using a straight line. However, we can do the following procedure. We can calculate the mean of variable 1, and we can calculate the mean of variable 2, and then we can draw a new graph where the x-axis is the distance of the point from the mean of variable 1, and the y-axis is the distance of from the mean of variable 2. This will transform the data to look something like this. And this is easily separable using this kind of line. So far, in the support vector machines that I've illustrated, we've only used two variables. However, the same ideas apply if you have more input variables. Instead of having a line, you end up with a surface that slices through the n-dimensional space. In the previous lecture, we talked about how we use the training sets to build many kinds of alternative models. Here, we can make this a bit more concrete. If the model type is a support vector machines, the alternative models will have the following differences between them. First of all, you can change how important you consider the accuracy versus the gap size, how far away the points are from the central line. You can also vary the model by varying the different transformations you can do before finding a line that separates the points. Next, we'll cover decision trees and random forests. Here's an example of a decision tree where the input variables are age and BMI, and the output variable is risk category. In a decision tree, you can learn a model where a series of yes or no questions are asked, and through that, you can make your way through the tree. So for example, if a patient is less than 60 years old, then the answer to this question is no. But if they're over 40 years old, then you go this way. If they have a BMI that's less than 30, then you go this way. And then you find out that this patient is low risk. In random forest, you can train a couple of different decision trees. Then, if you have a new observation, you can input all the input variables into each of the decision trees, and each of these decision trees will output a specific category. Then you can return the most common category returned by all of the different decision trees, basically like a voting system. In this case, the alternative models vary in the following parameters. First of all, you can vary the number of trees, and you can also vary the tree depth. The number of trees is the number of separate decision trees that are learned, and the depth is the number of yes or no questions that are asked by each decision tree. Finally, we'll briefly touch upon logistic regression. Logistic regression is useful when you have exactly two categories, yes or no, true or false. What you do is you fit a curve that looks like an S shape. What you do is you fit a logistic function, which is an S-shaped function that takes as input all of your input variables and produces an output that ranges between the values of 0 and 1. The closer the output is to 1, the answer is yes. The closer the output is to 0, the answer is no. Then we'll briefly touch upon variable importance. Once you have a model, you can ask, how important is each variable within the model? We're not going to go over how variable importance is defined in SVMs, random forests, etc. However, it's important to know which conclusions you're allowed to draw when you interpret the variable importance. For instance, you are allowed to say that variable A is more important than variable B in this model. You can also say, in model 1, variable A is more important than variable B, but in model 2, it's the other way around. You can also say variable A is useful for distinguishing between categories because it has a high variable importance in this model. However, you cannot necessarily say that variable A is more important in model 1 than model 2. This is especially true if the models are based on different algorithms, because variable importance is calculated in a different way for different algorithms, and it's not mutually comparable. In addition, you cannot say that variable A does not differ between categories because its variable importance is low. You could have a very big difference between the categories and still have a low variable importance because of redundant information. So the big takeaway messages is that we've learned about different ways of assessing model success, such as accuracy or the area under a ROC curve. We've also learned about different kinds of classifiers, such as support vector machines, which work particularly well if the data is linearly separable or can be transformed into being linearly separable. We've also learned about random forests, which are very good when there's nonlinear interaction between variables. And we've learned about logistic regression, which is particularly well suited for binary output variables. We've briefly touched on how to interpret variable importance and what you can conclude and what you cannot conclude.